Good afternoon to all the EAPCET aspirants, and today we are going to deal with the most major topic, that is plant growth regulators from the chapter Plant Growth and Development. Plant growth regulators is the key concept where in each year, if you take, in every year of NEET or EAPCET if you can take, there will be one question on plant growth regulators for sure. So, here if you see, plant growth regulators are also addressed as phytohormones, they are also addressed as phytohormones or plant hormones. What are these plant growth regulators actually? They are the small simple chemical components which are synthesized in any part of the plant body, and their function will be on the other end, like it can be on the synthesis region or over any other region of the plant body. Okay, these are small simple active chemical components, small simple active chemical components or chemical constituents synthesized in small concentration and given out to any other part of the plant body. Their mode of action can be anywhere in the plant body. As such, if you see plant growth regulators, they can either promote the growth and development of the plant, or they can inhibit the growth and development of the plant. As such, we have two types of plant growth regulators. One section is plant growth promoters and one more is plant growth inhibitors. Okay. Plant growth promoters include auxin, cytokinins, and gibberellins. Auxins, cytokinin and gibberellins. Plant growth inhibitors will be abscisic acid, that is ABA. Okay. So, you have plant growth promoters and plant growth inhibitors. Ethylene will do dual function. Okay. Ethylene will perform dual function both as plant growth promoters and as plant growth inhibitor. Clear, right? This is about plant growth regulators introduction. And now if you see all the phytohormones, what you have to learn from phytohormone exactly? What is the precursor and what are the derivatives of the every specific phytohormone, and then the discovery process, what are its physiological functions and applications of the phytohormone. These 5 segments you have to be clear with every plant hormone. If you look into this table clearly, for each phytohormone, you can see like phytohormone auxin. Auxin will be having IAA, indole compounds. Their derivatives are what? Indole compounds. And precursor of auxin is tryptophan, which is the aromatic amino acid. This is the precursor of auxin. And then you see cytokinin. Cytokinin whose natural form will be zeatin, artificial form of cytokinin will be kinetin. And these are adenine derivatives or purine derivatives. Purine derivatives in specific, the cytokinin is adenine derivatives. And this precursor, if you see, adenine is the precursor for cytokinin synthesis. And then gibberellins, Ga3, the chemical name over here is terpenes. Okay, these are the what are the constituents of gibberellins? Gibberellins are the derivatives of terpenoids. Okay, terpenoids or terpene derivatives. And here if you see the precursor, it is acetyl CoA and mevalonic acid. Acetyl CoA or mevalonic acid is the precursor of gibberellins. Next, abscisic acid. The chemical nature or the composition of abscisic acid is sequiterpenoid, that is 15 carbon terpenoid, and precursor is violaxanthin. Ethylene is a gaseous unsaturated hydrocarbon whose precursor will be methionine. Clear, right? These are the phytohormones, chemical compositions and their precursor. Chemical composition, you can also address this as derivatives of the phytohormones. This table is very important, ma. Take a screenshot, revise it multiple times. You can see whole lot of PYQs on this. Very important. Fine. Now, we will exercise each hormone one by one. First, we will move on with auxin. Okay. Auxin. Who discovered auxin? It is first by Charles Darwin and Francis Darwin. Charles Darwin and Francis Darwin, they have synthesized, they have discovered this hormone by first analyzing or identifying phototrophic movement. On what seedlings? Phototrophic movement by coleoptile seedlings of canary grass. What is the coleoptile in monocots? If you have observed the monocot seed, over the embryonal axis at the tip, plumule will be present. Plumule is covered by a protective sheet known as coleoptile. This coleoptile, when extracted from canary seedlings and when unilateral light is placed over it, when this is subjected to, when coleoptile, canary grass seedling coleoptile is subjected to unilateral sunlight, it has showed bend towards the sunlight like this. Bent is shown towards the sunlight, which is showing phototrophic movement. And later F.W. Wendt has isolated auxin from human urine. Okay. F.W. Wendt has isolated auxin from human urine. One more characteristic feature. F.W. Wendt has conducted sequential experiments after analyzing the phototrophic movement of the coleoptile from canary grass. He has repeated the experiment by taking the coleoptile of oat seedlings. Coleoptile of oat seedlings. So, here if you observe, when coleoptile of oat seedling, when it is dissected and it is placed over the agar block, when it is placed over the agar block, when the coleoptile is dissected and kept over the agar block, the molecules, the chemical constituents, they travel all across and they will reach to the agar block. This agar block is filled with all the constituents which are being extracted from the tip. So, over this dissected region, when agar block is placed, and it is subjected to the unilateral light, it has shown a bend like this, okay, which means that at the apical regions, the constituents which are rich are auxin. Auxin is rich in the apical regions, both at the shoot apical meristem and at the root apical meristem. In the apical regions, the auxin which is rich, it is travelling across so that it is showing the bend or the movement towards the sunlight. So, it is the first auxin discovery. What you have to remember, ma? First come first about the experiments are done on the coleoptile of canary grass by Charles Darwin and Francis Darwin. Second is by F.W. Wendt, who has isolated auxin from human urine, okay, and he has performed the experiment on coleoptile of oat seedlings by taking the agar block, and he said that these chemical constituents will travel across to give out the phototrophic movement. Clear till here? You have to remember these three, very important. And now if we see the functions and applications of auxin. The first and foremost function of auxin is apical dominance. What is this apical dominance? So, just now we have observed auxin is rich in, auxin constituents will be high in both shoot apical meristem, root apical meristem. In the apical regions, the concentration of auxin will be high. So, it will promote the growth of, apical buds, and it will inhibit the growth of lateral buds. It will promote the growth of apical buds and it will inhibit the growth of lateral buds. Fine, right? That is about apical dominance. Second is cell division. So, apical dominance, auxin will promote apical dominance by promoting the growth of apical buds and inhibiting the growth of lateral buds. Second is cell division. Auxin will promote the cell division and it will promote the abscission of older leaves and older parts of the plants or older fruits, and it will inhibit the abscission of younger leaves. It will inhibit the abscission of younger leaves and it will promote the abscission of older leaves. Next is xylem differentiation. Xylem differentiation is also done by auxin itself. So, after knowing functions, you have to know about two types of auxins which are very important. Those are two types of auxins which are important. First will be natural auxins and second is synthetic auxins, you have to know. Okay. So, two types of auxins. Natural auxins and synthetic auxins. Natural auxins include indole acetic acid and indole butyric acid. Fine, right? Synthetic auxins include naphthalene acetic acid and 2,4-dichlorophenoxyacetic acid. Synthetic auxins include 2,4-dichlorophenoxyacetic acid. And if you see, auxin promotes rooting in stem cutting regions. This is promoted by naphthalene acetic acid, okay, naphthalene acetic acid and indole butyric acid. Okay, this will promote rooting in stem cutting regions. Next is flowering. Second application will be flowering, and this is promoted by again two synthetic auxins. First is 2,4-dichlorophenoxyacetic acid and naphthalene acetic acid. This promotes flowering, especially in pineapple. Fine, it promotes flowering in pineapple. And then if you observe parthenocarpy, that is production of seedless fruits, that is fruit formation without fertilization. Without fertilization. This is promoted by indole acetic acid and indole butyric acid, both are natural auxins only. And auxins will promote weed-free lawns and they act as weedicides. Few of such synthetic auxins include 2,4-dichlorophenoxyacetic acid and 2,4,5-trichlorophenoxyacetic as well. Okay, so they act as weedicides and they produce weed-free lawns. Fine, clear till here? So functions include apical dominance, cell division abscission of older parts of the plant, and prevention of abscission in the younger parts of the plant. And it includes xylem differentiation, and if you look into the applications of auxin, applications of auxin include rooting, flowering, parthenocarpy and weedicides. Of this, most important segment you have to remember is NAA and IBA, one of which is natural auxin and the other is synthetic auxin, which will promote rooting in stem cutting regions. And next, flowering, especially in pineapples, it is promoted by 2,4-D and NAA. These are the synthetic auxins. And parthenocarpy, which is production of fruits without fertilization, this is promoted by natural auxins like IAA and IBA, that is indole acetic acid and indole butyric acid. And last application of auxin is as weedicide, that is producing weed free lawns, especially it will eliminate dicot weeds. By 2,4-dichlorophenoxyacetic acid. Next is cytokinin. Cytokinin is also plant growth promoter. Plant growth promoter. Fine. So, cyto, which means cell, kinin, which is movement. The name of the hormone itself is saying out. The main activity that cytokinin performs is cell proliferation and nutrient mobilization. And here you have to know both the natural and artificial cytokines. Natural form of cytokinin is known as zeatin, and cytokinin, the constituents can also be extracted from coconut milk, yeast extract, fine, right, from coconut milk, yeast extract, the cytokinin constituents can be extracted, and the natural form of cytokinin is zeatin. Artificial form of cytokinin is kinetin, which is extracted from herring sperm's DNA. Herring sperm DNA. Fine, right? Clear? Zeatin and kinetin. Any doubts anywhere? Zeatin and kinetin, both natural and artificial forms of cytokinin, and the main activity is cell proliferation and nutrient mobilization. How come the cytokinin is being discovered? Along with auxin, when cytokinin constituents, that is the extract from coconut like coconut milk and yeast extract, when it is supplemented to a callus, they have observed extreme cell proliferation and increase in internodal elongation of tobacco callus, increase in internodal elongation of tobacco callus. Fine, in the test tube, when cytokinin is added along with auxin, increase in cell proliferation is being observed. So, from this you have to remember this, okay. Miller et al. has isolated cytokinin from herring sperm's DNA, and you have to know what is the natural form and from where it is extracted, like from coconut milk and yeast extract. What is the main activity? Cell proliferation and nutrient mobilization. Now, we look into the functions of auxin. First is cell division. It will promote the active cell division. Second, that is delay in senescence, which is shown by Richmond-Lang effect. fine, and this overcomes apical dominance by promoting the growth of lateral buds. This promotes the growth of lateral buds, that is how it overcomes the apical dominance. Next is growth of shoot and growth of roots, and you can also add on one more function, that is nutrient mobilization. And then if you see applications, first is in tissue culture. In tissue culture, how cytokinin is being applied? For example, just now we have observed the cell proliferation is increased in the tobacco callus and internodal elongation has been increased by the application of cytokinins. So, here if you see first in tissue culture, equal concentrations of auxin and cytokinin. Let us suppose 1:1 concentration of auxin and cytokinin. This will promote callus formation. Fine. And then if you see auxin and cytokinin, more auxin concentration and less cytokinin, this will promote root growth. And less auxin, more cytokinin will promote shoot growth. Fine. So, 1:1 is for callus, 2:1, that is more auxin, less cytokinin will promote root growth, and more cytokinin, less auxin will promote shoot growth. Clear with functions and applications of cytokinin? Any doubts anywhere, ma? Now, we will move on to the next phytohormone, that is gibberellic acid. Gibberellins is being identified by a fungal disease in Japan, and this is identified by the scientist Kurosawa, okay, which led to the fungal disease which caused by Gibberella fujikuroi. Gibberella fujikuroi has caused an abnormal increase in the length of the rice plants, abnormal increase in the length of rice plants. This internodal elongation and increase in the length of rice plants is later observed by the constituents that are secreted by this fungus, okay, by the constituents which are secreted by this fungus, where it has released the constituents like gibberellins, which has increased the internodal elongation, and thereby the rice plant length has been increased, is what identified by the scientist Kurosawa. So, the first discovered gibberellin is gibberellic acid 3, and the pH of gibberellic acid will be low and its nature is acidic. It is first discovered and widely used. Widely used. Fine. And then if you observe next, functions and applications of gibberellic acid. Functions, you see, gibberellic acid, it promotes seed germination, and here for auxin and cytokinin I have to tell you on synergistic and antagonistic effects. The synergistic effect, that means both the hormones performing the same function, they have the same physiological role. Auxin and cytokinin are synergistic in the effect of cell division. They are antagonistic in the effect of apical dominance, wherein auxin is promoting the apical dominance and cytokinin is inhibiting the apical dominance. Clear till here? Any doubt anywhere? Synergistic and antagonistic effects are very important. Seed germination, gibberellins will promote the seed germination. How gibberellins will promote the seed germination? In seed, let us take a monocot seed only, okay, where it has, if you observe a maize seed, it has two types of endosperms, amylose and protein endosperm. Starchy endosperm and proteinaceous endosperm. Two endosperms will be present. In the starchy endosperm, the starch has to be simplified for the absorption or for the growth of the embryo. Okay, it has to be absorbed properly. Okay, for that the enzyme amylase is required. Amylase is required, which will oxidize starch to simple sugars, so that it will be absorbed by the embryonal axis and it will promote the growth of the seedling. It will promote the growth of the seedling. So that is how gibberellic acid will promote the synthesis of auxin. Gibberellic acid will promote the synthesis of amylase. This amylase will start acting on the starch. It will simplify starch to the simple sugars, and that is how it will be helpful for the growth of embryonal axis. This is how gibberellins are performing seed germination activity. Second is bolting. Bolting is nothing but spraying gibberellins just prior to the flowering. Spraying gibberellins just prior to the flowering will increase the internodal elongation of the floral stalks. Okay, of the floral stalks, the internodal elongation will be increased. Thereby, post fertilization, a completely occupied fruit or vegetable is formed, which is full sized fruit or vegetable. Okay, so spraying gibberellins prior to flowering will increase the internodal elongations of the floral stalks, and post fertilization this will help forming a full sized vegetable or fruit. This is being observed in rosette plants. Rosette plants. Next you observe is stem elongation. Stem elongation is that is internodal elongation is observed by gibberellins. Best application you see in sugar canes. Massive yield of sugar canes will be increased by the application of gibberellins, because it will increase the internodal elongation. Fine, right? So, seed germination, bolting and stem elongation, these are the major functions of gibberellins. Gibberellins are widely used in brewing industries because they enhance the synthesis of amylase. Okay, they are widely used in brewing industries for malting process. Next, it will enhance the sugar cane yield, and spraying gibberellins on conifers will yield quick maturity, and gibberellins will delay the ripening process and it will promote the fruit growth also. When the floral stalk is increased, the fruits get better shape. The fruits get better shape. For example, apple after spraying the gibberellins, and apple when gibberellins is not sprayed. This much variation is observed. Fine, right? So, these are the functions and applications of gibberellins, very important. Keep noted with it. And after this, the next phytohormone we are going to see is abscisic acid. Abscisic acid, which is popularly known as stress hormone, or anti-transparent hormone. Abscisic acid known as stress hormone or anti-transparent hormone. What does this do? Abscisic acid. Abscisic acid will be synthesized in the stressful conditions, where it will promote the growth of the plant even in stressful conditions. For example, in desert areas, whenever there is heat stress, abscisic acid will induce the stomatal closure, okay, and it will promote the abscission of older leaves and younger leaves also, okay. During stressful conditions, when plant is unable to hold the bulk of the leaves and fruits, then it will promote the abscission of young flowers, fruits, buds and leaves as well. And it will promote seed dormancy during unfavorable conditions of the plant, and it will inhibit the metabolism. So, in inhibiting metabolism, this is antagonistic to cytokinin, where cytokinin will promote the metabolism. And with respect to seed dormancy, abscisic acid is antagonistic to gibberellic acid, and with respect to abscission, it is antagonistic to auxin. Auxins will inhibit the abscission of younger leaves and fruits, whereas abscisic acid will promote the abscission of younger buds, leaves and fruits. Next, applications of abscisic acid. Dormancy, abscisic acid will promote the dormancy, and this acts like an anti-transpirant. Clear till here? Any doubts anywhere, ma? And then last hormone we are going to see is ethylene. Ethylene is the phytohormone which has dual functions, both as plant growth promoter and plant growth inhibitor. So, this is first observed by Auxins, wherein he has observed that ripened oranges, when they are placed beside the unripe mangoes or bananas or any other fruit, they got ripened too. Okay. So, there is some gaseous enzyme or gaseous hormone which is being secreted from oranges, which led to the ripening of mangoes or bananas. Okay. So, ethylene here, ethylene is a gaseous hormone which has enhanced the ripening of other fruit. Fine, right? Auxins, who discovered that? Auxins. And in recent applications, we use ethylene in the form of ethaphon for fruit ripening. After this, let us look into the functions of ethylene. Ethylene will promote the flowering and it will lead to increased fruit set. This is the synergistic effect of auxin 2. Example is pineapple itself, and it will promote the fruit ripening and triple response is observed. That is, it will increase the girth of the axis. Axis girth will be increased and it will promote horizontal growth of the seedling, and it will lead to apical hook formation of the seedling. This is the triple response which is caused by the ethylene. That in the rice seedling it is observed that it will increase the width or diameter of the main axis of the seedling. Second thing is it will promote the horizontal growth, and third is an apical hook formation is observed. This is the triple response which is shown, and it will increase the senescence. Senescence is promoted by ethylene, and adventitious root formation is also observed by ethylene. And here if you see in the applications, of course we will use ethylene as ethaphon for fruit ripening, and abscission and thinning of the fruits, increase in fruit set, specially observed in pineapples, and sprouting of storage organs is also observed. So, at the end, if these are the functions of ethylene, at the end if you have to consider both the synergistic and antagonistic effects. Synergistic and antagonistic effects. Synergistic effects which will promote cell division, those which promote seed germination, cell division is by auxin and cytokinin. Seed germination by gibberellic acid and ethylene. And flowering What is the synergistic effect by two phytohormones? Auxin and ethylene. These are the synergistic effects. Antagonistic effects, if you see, with respect to seed germination. Abscisic acid will inhibit the seed germination, whereas abscisic acid will inhibit the seed germination, whereas gibberellic acid will promote the seed germination. This is the first antagonistic effect. Second is cell proliferation or cell division, or you can observe apical dominance. Okay, apical dominance. With respect to apical dominance, we have two antagonistic effects, whereas auxin will promote the apical dominance, cytokinin will inhibit the apical dominance. Next, seed germination. Yeah, seed germination is already done. Next you can take is abscission. Of younger leaves. It is inhibited by auxin and abscission is promoted by abscisic acid. And last, if you consider senescence, promoted by ethylene, inhibited by cytokinin. Clear till here? Any doubt anywhere, both about synergistic and antagonistic activities? That's all with plant growth regulators. Any doubt you are having in this, you can ask me in our next session. We are going to solve the complete EAPCET previous year questions from all the chapters of plant physiology. So come prepared for the next session.